Welcome to EXO Beyond the Cells podcast. We're here to explore the frontiers of cell communication, how cells interact with each other, and the biology beyond the boundaries of the cell. I'm Brent Stockwell, editor-in-chief of EXO Beyond the Cell. The journal focuses on publishing high-impact research on cells interacting with their environment, and that spans extracellular biology, spatial omics, and the complex dynamics of tissue environment. In this podcast, I'm speaking directly with our authors about the ideas shaping their research and their discoveries that are driving this field forward. For today's episode, I'm joined by Professor Shinya Toyokuni and Dr. Yingyi Kong from Nagoya University. They've spent years studying oxidative stress and cancer and how iron and environmental exposures like asbestos can drive disease. Today, we're going to talk about their recent article in EXO entitled Ferroptosis in BRCA-associated disorders, extracellular vesicles as potential messengers beyond the cell. This article tackles a fascinating question. Can ferroptosis spread from one cell to another? And if so, how? Their work looks at how extracellular vesicles carry these signals, shaping how cell death affects tissues more broadly. Professor Toyokuni and Dr. Kong, it is a true pleasure to have both of you with us today. Thank you for coming.
Yeah, that's weird. We are very honored to be invited for this interview. Yeah.
Thank you.
Thank you.
Invitation.
Thank you. So let's get into talking about your paper. So most people know BRCA genes are linked to cancer risk, but what's the new idea that your paper introduces about these genes and how they can affect cells?
Yeah, it's a great honor to talk to you. You coined the word ferroptosis, and actually we have been working on this iron excess-induced carcinogenesis for more than 30 years. And there was not much study to see the relation between the ferroptosis and any genetic diseases. So like 10 years ago, we decided to tackle with certain genetic diseases. So we have chosen BRCA1 and 2 genetic disease because this is very closely associated with carcinogenesis and also has certain social impact. However, of course, this gene was discovered in the 90s, and many mice model was produced starting from that time. But having 10 or 20 years, it was found that mice models, actually knockout mice, does not show the phenotype of humans. So we decided to do other animal models, namely rats. And this model shows very interesting and results. And we found that in this model, maybe later Professor Kong will talk more in detail, but in BRCA1 knockout rats, iron-induced carcinogenesis is significantly promoted. And the important point we found is that both in BRCA1 and BRCA2 knockout rats, iron metabolism is impaired at least in renal cells. That's quite interesting. That's even in the normal status, mitochondrial activity and also morphology is greatly impaired in those hetero knockout rats. And that caused the dysregulation of iron. So this has not been pointed out thus far, and this is quite interesting, we think.
That's great. There's a couple of very interesting things you said there. So let me ask, about the rats versus the mice. So why do you think you get different effects in rats versus mice when you regulate BRCA genes differently?
Yeah, it's of course it's a little difficult to answer, but of course that's the species difference and their lifetime length is different, and we have to search it in the near future, I think, but there's certain difference. So far, we know that also the events of carcinogenesis is quite different between mice and rats. Rats shows more aggressive cancers. That's quite similar to humans. But in mice, we see very subtle change and less aggressive tumors. So at present, we can just say that species difference, and it's actually a difficult question for us at present.
Dr. Kong, I have a question for you, a follow-up about BRCA1 and BRCA2. So in the paper, you talk about different effects of BRCA1 versus BRCA2 on ferroptosis sensitivity. Can you tell us a little bit about that and what's the difference and why you think there's a difference?
Yeah, sure. Yeah, actually, the most like a traditional view of the BRCA study is about the DNA repair, but there's another like a consequence point of the BRCA mutation has been like ignored for a long time, and actually, yeah, my major work is about the BRCA1, and we found like a partial escape from the ferroptosis after the our interaction-based carcinogenesis model. But for the BRCA2, actually the ferroptosis has been executed after our like oxidative stress stimulations, and those promotion for the renal carcinogenesis has been revealed in the BRCA1 mutated model, but not in the BRCA2 mutated animal models. So that been quite interesting, like how that happened and how that could lead to the different outcomes of the studies. And also we observed the c-MYC amplification in the BRCA1 mutated generated renal carcinoma, which is actually synthetic to the high-bulk breast cancer patients. So we believe that's like a strong point that our rat model has like mimic the human phenotype.
Interesting. So, and do you have any idea why BRCA1 and BRCA2 are having these different effects?
Yeah, our first point is the BRCA1 is most like a sensor for the DNA damage. So once that caught those signals that could like connect it to other compounds and leading to the further repair prevention. But for the BRCA2, that's mostly effect in the DNA damage repair. So that's like for all the eukaryotic cells, the BRCA2 is essential for their like the DNA repair progression.
Interesting. Professor Toyokuni, let me ask about the extracellular vesicles that you talk about in the review that can propagate ferroptotic stress and allow this communication between cells. So can you tell us a little more about that, how that works and why that's important?
Yeah, this paper was we published about 4 or 5 years ago, but it's a new finding about the ion metabolism. It's quite interesting, and we found ion responsive element sequence in CD63. You may know that there are several markers for exosome. That's CD9, CD63, and 81. but only CD63 of humans showed IRE sequence. That's a sequence that's specified for the iron metabolism genes, like a transferrin receptor, DMT1, ferropotin, like that, and ferritin as well. And from that discovery, we found that those iron metabolism may be associated with the exosomes. And what we found is that if certain cells have enough amounts of iron, of course, that is stored as ferritin. And that ferritin is exported, that's packed in exosomes. Finally, to other cells that is deficient in iron. So they're sharing those excess iron between autocells. And that's fine. More interesting thing is that the IRE sequence was found in only in higher primates, not in experimental rats and mice. That's quite interesting for us. So this system is probably working for the evolution we are thinking. And that's for the normal cells. But for more pathological status, like in asbestos-induced mesothelial carcinogenesis, we have been working for that maybe for 15 years. And this is also carcinogenesis associated with excess iron. And in that case, asbestos is a foreign body. So the phagocyte, like macrophages, try to engulf them and try to dispose them. But they can't, and they go into ferroptosis. That's quite interesting. But at the time, very specific exosomes with labeled with CD63 are secreted. And unfortunately, the target cells or mesothelial cells catches those CD63 ferritin-filled exosomes and will be iron loaded. And we found that that was coexistent with DNA, double strand breaks, and other base modifications. So this is actually a quite new molecular mechanism, one of the mechanisms for asbestos-induced mesothelial carcinogenesis.
Fascinating. So do the exosomes that are released in that case by the macrophages have asbestos in them as well?
Yeah, released from the macrophages, and that is received by the target cell, cancer target cell, mesothelial cells.
And is asbestos found in the extracellular vesicles as well as in the macrophages?
The size of the asbestos is they're huge, considering about the single cells actually. The length is like 100 to 200 micrometer, but the width is quite small, like 20, less than maybe less than 10 micrometer. So phagocytes try to engulf them. That is possible, but the membrane is ruptured, and finally they got into ferroptosis in that case. And the interesting point is that the asbestos during our body absorbs a lot of hemoglobin, so collecting iron on the surface of asbestos. So different lengths of asbestos are present in our body. So they are collected in phagocytes, but they engulf a very long asbestos. They die via ferroptosis.
Got it. Okay. Interesting. And do you think ferroptosis therapies that block ferroptosis could be beneficial in mesothelioma to block that propagation of iron?
That's possible, I think. Of course, that's possible. That of course, the first point is not exposing to asbestos is most important, of course. And this process is taking quite a long time, like decades, from inhaling, from the inhalation of asbestos fibers that reaches trachea and lung tissue, and that should go through the lung parenchyma. That takes 20 to 30 years. So avoiding those exposure is most important. But yeah, those stopping ferroptosis may work as a prevention. We have to think about that.
Great, great. Okay. Dr. Kong, let me ask you another question about the paper. You talk about an evolutionary argument in the paper about how BRCA mutations might have been beneficial in the past. Can you tell us about that idea?
Yeah, actually, I'm not the expert, but yeah, Shinya is the expert for evaluation. But from my point of view is that the BRCA1 or 2 are like quite common in the human populations, even after the maybe thousand more than that years of the evaluation. So we thought that there's a possibility that that those kind of mutation remain is because so in those like ancient times, the food and the environment was quite different from now, and those times people I like hunting for food or like struggle really hard to get the food. So they're being through some food like inefficient, and also the iron deficiency are quite common at that age. And so in those kind of environments, we believe those like the cancer risk like are like less important than the like the living issue. So instead, those kind of survival in that such kind of environment need those kind of mutations for human beings to like execute the cells by ferroptosis and other procedures and keep their maybe the like the cellular metabolism running in like a low cost. But now the environments are quite different, and the risk for cancer and like other diseases associated with those maybe the nutrients like over consumed, and we believe those kind of situation like current situation may bring up our attention for the those kind of strategies to prevent human from the diseases that is maybe being like susceptible because of those mutations, especially for the BRCA1 or 2.
Got it. Okay, that's great. Let me just end today's podcast. I have one more question for each of you. I think, first of all, I want to encourage our listeners to read your paper because it's an excellent article and very interesting and touches on these ideas we've talked about today. But for my last question, I want to ask each of you, what are you excited about in the future? What's the most exciting thing that's in science that you're working on or thinking about for the future? So, Professor Toyokuni, do you want to start?
Yeah, of course. The regulation of iron is very important, I think. No species on the Earth can survive without iron at present, I think. So currently, we have overcome a lot of infectious diseases and a lot of problems, and the lifetime is getting longer and longer. So during aging, most of us know that there'll be the overdose of iron. Actually, we have accumulated too much iron, so that means that the regulating this iron is quite important, and the one of the the other most important principle for iron metabolism is that we cannot excrete iron to outside of our body unless there's some bleeding, actually. This is a semi-circular system, and a very small amount is taken up and excreted. We have around 4 grams, 4,000 milligrams, and 1 milligram in and 1 milligram out. So the regulation of iron is quite important. And currently, we are working on iron chaperones inside cells, and whether we can control those iron in comparison to according with the aging would help to increase our health condition, we think. So some of the molecules are working as iron chaperone inside the cell is quite exciting for us at present. And maybe we can kill the cancer cells with new modality. I think I wrote a little like low temperature plasma that actually attacks the presence of those iron. Cancer cells needs iron because DNA replication needs iron. So cancer cells are collecting iron. That may be the Achilles heel of cancer cells, we are thinking. So we are quite excited on that and trying to find new modality for cancer therapy.
Very exciting. Very exciting. Dr. Kong, do you want to share something you're excited about for the future?
Yeah. Actually, by studying the BRCA1 associated study, I found there substantial possibilities for the future therapeutic options for the high bulk patients, especially for those carriers of BRCA1 or 2 mutations often occurring for the triple negative breast cancer. But for the treatment, although we have the options for those PARP inhibitors, still the options are quite limited. And we expect that by studying into the BRCA1 or 2 and their connection with the EVs could produce a new system to the earlier prognosis or the further therapeutic strategies for those high bulk patients in the perspective of ferroptosis and other iron associated mechanisms, and we believe that's quite excited for the cancer therapy and as the perspective from the fundamental researchers.
Fantastic. Great. Well, I want to thank you both for joining us today and telling us about your article. Yes. And goodbye to our listeners. And we hope to see you in the future on the next podcast. So thank you, everybody.